So thank you, Thomas, and Arunima for giving me the opportunity to talk. Unfortunately, I had to skip at the last moment for not able to be on site. But I'm happy to chat about to discuss some of the work that we have been doing from the atmospheric science point of view, and how multimodal chemical imaging has been uh, used in our research. And I'll highlight a little bit more, some of the parts that we're gonna talk about, like how this thing can be improved in the future. So I'll give some perspective to that as well. So before I start, I just want to make sure we have a large team members that has been behind this work that I'm going to present within PNL, and we also have several external collaborators that we are working with very closely to understand or use the chemical imaging to understand some of the atmospheric processes. And specifically, I want to shout out for these folks that are kind of doing a lot of the heavy lifting for doing all the work for under chemical imaging or atmospheric science in general. So really thanks to them for doing all the work. I know this is not an atmospheric science specific background, so I just want to give a very brief introduction about why we study atmospheric particles and their impact on the climate. So this is a very nice illustrative view that I little bit modified it from a review paper. Kind of give a representation why this is important and different component of our system. So the main story of this slide here is talking about the aerosol as a linkage between different Earth system. So for example, particles can emitted from the land, and there are some soil and dust particles that have a micronutrient such as iron-containing particles can deposit over the ocean and that act as a nutrient. And similarly, from particles that are emitted from the ocean or lake surfaces can travel long distances and deposit on the land or the plants. And then there's and biogenic emission from plants can again get into the atmosphere and interact between the different part of the ecosystem. And it can affect the deposition of the particles, photosynthetic effect, transport of nutrient. And for atmospheric component part that we're really interested in is the visibility and the radiative forcing, meaning that how atmospheric particles interact with the sunlight and affecting the radiation budget, and also affecting the interacting with the clouds and how they affect the solar energy. So as you can see, some of the atmospheric particles, they are indeed very complex in nature. And again, this is kind of a backup from the previous slide that kind of really show that, as I mentioned, there's this different component of the ecosystem interact between them and also with the atmosphere. And then if you look at from this ecosystem to the individual particles, they can have in the different phase, meaning that they can exist in the solid, semi-solid or liquid state, and they interact constantly with the gas molecules. So they have this molecule and cellular interaction happening, and we're trying to understand these processes that can have a multi-scale, so from kilometers to nanometer scale, and they have a multi-phase processes. So that's why we need a multimodal approach to understand these atmospheric particles and understand their impact on the climate. Here we specifically use defined modality of microscopy, spectroscopy, and mass spectrometry to understand the atmospheric processes. There are varieties of microspectroscopy and mass spec available, for example, starting from optical that can be incorporated, for example, Raman microscopy, and also that atomic force microscopy, and then here we're talking about the scanning electron microscopy and transmission electron microscopy, and so on. So I cannot list all of them. So I'm going to focus a few of those that we use more frequently, at least in our research. But I just want to mention that there are a bunch of different this modality that has been used in the atmospheric science, and there are different pros and cons. But the main point here is that when we use this method as a combined, we can answer better some of the challenging questions that we're trying to answer for atmospheric science. So Odeta already gave a very nice introduction about the scanning vector microscopy, so that make my life easier. So I don't need to explain how the principle it works. So briefly, I'm just saying that how we use for our atmospheric science program. So for example, if you look at the individual particles, and you can get X-ray spectra for each of these individual particles. And the way we do, as Odeta mentioned, is a particle feature detection method. So we can automatically detect the particles, and the microscope can run in the autopilot mode, and you can get thousands of particles analyzed. So once you have all this information of particle size, the morphology, and the elemental composition, and then from this elemental composition, different fraction of these elements, you can group them into different particle classes, and then you get a size distribution that are chemically specific. So then we can understand what are those, different types of particles that are present in the atmosphere. And then here is just one example. You can see that you have the different particles. For example, is this one particularly showing them in Amazonia rainforest at nighttime and daytime. We're looking at the above and below canopy. And one thing we surprisingly found is that 50% of those particles are sodium-rich particles. So I'll briefly mention about this story a little bit and why the multimodal imaging really helped us to understand this puzzle here. And to do that, I have to introduce on one method here, which is we often use, and that's why I am at currently at the Advanced Light Source at the Lawrence National Laboratory, where we use a method called scanning transmission X-ray microscopy with a near-edge X-ray absorption fine structure spectroscopy, short name STIXM. So here is a exact X-ray absorption-based technology, so you can raster scan your samples, and after incident soft X-ray, you can look what are the after interacting with the particles, what are the transmission, and measure with the X-ray detector. The beauty of this instrument that can give you a functional group level information at the individual particle level. And again, I'll explain why we use that and the complementary to some of the scanning electron microscopy we use. So we often use the electron microscopy and the X-ray microscopy to answer some of the questions that we cannot answer just with the one technique here. So coming back to my story about this puzzle that we have with that, this lot of sodium-rich particles in Amazonia, and when we found, look at the specific particles, this looks like a fungus spore particle, and they have a sodium peak in those. But the problem is this with the EDX that sodium and copper are very close by, and we use a copper TM grid to collect those particles. We need an independent method to verify that these are indeed sodium containing particles. So then we use to the STIXM, and this is a high energy STIXM, which sodium energy is kind of 1000 eV. So and this is the same particle you can see, and then we observe that indeed these particles contain sodium. And then we also still wanted to make sure that, whatever their distribution, then we looked into their these particles under nanosims, which again explains some of the previous talk before. So nanosims is very good for detect sodium because of their higher ionization potential. And then a scale is show that, these particles indeed contain sodium. So that's kind of really give us the confidence to understand that, what are the sources of these particles. Not necessarily these are, if you think about sodium, you expect that these are coming from a C-square also, but indeed here is a different scenario here. So this multimodal imaging really helped us to understand that process or source of these particles. So here perspective is that really we need a multimodal imaging, but the one that the work we did here is really manually try to identify this, locate these particles. We need a more advanced particle tracking features between different instrumentation that we use. We need a power adapter and some smart way to do it. And also in some cases, we need a broader energy range. For example, sodium has a higher energy range, so we cannot use just a traditional soft X-ray, which is covered until like 750 or something. So these are kind of, for multimodal imaging, we need some advancement automation that Odeta was mentioning in her talk. So going back to about these biological particles, we kind of looked into this their pattern and their cloud formation potential. So we picked these two particles, so not all the biological particles contain sodium. So the below one is sodium containing particles, top one is non-sodium containing particles. And as you increase the relative humidity, you can see the sodium containing particles, they go uptake water and quite become, they have their growth, but the sodium containing non-sodium containing particles did not uptake water. So that's kind of very reflect that the composition also very important to understand their interaction with the water vapor. And so this give you the area growth factor, but for atmospheric modeling, you need a mass. So to do that, we use a STIXM, in situ STIXM. So here we are looking at the oxygen map. And again, this is an example of non-sodium containing particles. Even after 96% humidity, we did not see any noticeable changes. But for this upper particle, which is contain sodium, we observe significant changes. And from these images, we can get a mass growth factor, which can be used in atmospheric models that use a mass in most of the cases. I'll keep talking, and the reason behind this talk is kind of showing again the perspective I'll explain in a few minutes. That when you keep increasing the humidity more, and sometimes you go into this cloud cycle, meaning that you go increase the humidity, decrease it, and again increase the humidity. So going through these cycles, you see these particles start fragmenting and release these small tiny particles, and that become very important for atmospheric science, because these small particles can travel for long distances and stay there for a longer time. And then if you look at the sodium map here, you can see this hotspot of the sodium fragments within these individual particles. But the way we have been doing this experiment is that you blow them up to in the environmental SEM, and you dry them again, then image them. So we really don't see these particles in a real time. And to do that, we really need some in situ cell, even like X-ray microscopy in situ cell, but more advanced than the one that we used before, where we can have a cloud cycling. Also need to improve in terms of the imaging timescale, because some of the processes are much faster. So with the traditional timescale that we have, we were not able to capture those events. We need to also have the flexibility to do the gas flow. So sometimes they react with ozone or some other oxidant, and to try to see the interaction happening between all these environmental components. And then lastly is something easy to install and operate. The one that I've been using that has quite challenging, needs a lot of skill set to perform this experiment. But I believe that there are a lot of things can be make it simpler, and it will be easy to install and easy to perform this experiment in a more sophisticated way. So definitely there are a lot of improvement is needed. Going to the little bit a different topic now. Again, my main motivation here to show some examples where we use this multimodal imaging for different atmospheric science question. So this one was studied by Suzanne. I was a PhD intern here at PNL. While she was looking into these long-range transported biomass burning particles, a very specific type of spherical particles called towers in the atmosphere. And these are the elemental mapping from scanning TEM. And you can see some of the particles are very homogeneous in nature, while some other particles, possibly more aged, can have some inclusion of sulfur and potassium. So we try to understand this. Then we use again STIXM, where we can see there are two different type of particles, where you have more homogeneous, like I was mentioning, and some cases you have those elemental carbon. And then we use another spectroscopy in scanning TEM, which is energy loss spectroscopy. And then the very simple way to explain this is this energy loss spectroscopy can extract information for refractive index of individual particles. And once you get the refractive index, you can calculate their scattering and absorption, and then we understand how they're affecting the interaction with the sunlight. So that's kind of study shows again the highlighting different multimodal imaging can help to understand the optical properties of our source. Switching back to another topic here, this is again led by Noor Nahar at PNL, and what she was looking into this Arctic particles that goes into the upper atmosphere. So they the stratification of Arctic atmosphere, not only at the ground, but at aloft. And she was looking into these particles and both in the SEM and STIXM, and these are again same particles. Correlative imaging kind of shows this organic coating around these particles, which often indicate the cloud processing. So she was using this as an indicator and try to understand that at different layer of Arctic atmosphere, how this particle gets processed within the cloud or above the cloud. So that was kind of the main story that she performed. Again, switching back to another work that I really want to highlight also, that how you talk about I was talking about the STIXM and SEM quite a bit, but how these two methods can be combined and provide a more integrated product. So this one was led by Matthew Font and Ryan Moffett. And what they did is they look at the STIXM images, and STIXM is very good into the lighter elements. So, for example, if you're looking at the atmospheric particles, carbon, nitrogen, oxygen are very one of the key elements. So, and these are very good in more quantitative information from the STIXM, while the SEM ADX are very good for looking into the higher elements starting from atomic number 11, which is sodium. And then you automatically track these particles with the software they developed, and then you can look into the individual particles. Then what they did, they combined the mass of individual particles by combining carbon, nitrogen, oxygen from STIXM, and then use this anything elements higher than sodium from SEM, and develop this mixing state parameterization or mixing state model where you can quantify the mixing state of particles combining these SEM and STIXM. So that's like a really advancement in the field that they did, and that will help us to understand or quantify the mixing state of particles, because atmospheric particles often are multi-component, and it was really important to quantify the mixing state using these two approaches. The next one, I talk about the phase state of viscosity. This is something, another important topic in terms of atmospheric science that in the community we are trying to understand. When you talk about a phase, as I mentioned, they can be present in solid, semi-solid, liquid. And one way we can understand is that you have a solid particles impact on a surface. On a surface, you can have a particle shape did not deform, while you have a liquid or semi-solid particles, they deform their shape. So, and these particles can be lower viscosity or compared to one that have higher viscosity, maintain their spherical shape. And previous study shows that the lab-generated secondary organic aerosol, they can be more like a liquid state. So basically, if the slope is lower, that means these are liquid compared to the field-generated field particles. This can be in the solid state. And so we use that surveyed by Jay, where you see he's using the atmospheric particles from the ACM imaging from the tinted view, and then try to understand from the STIXM also to understand basically the total carbon absorption, basically slope of these curves to understand the viscosity. I know I'm running out of time, so I'll go faster now. One study that I want to highlight again very briefly, that again the essence of multimodal imaging, where we use the work made by Greg actually here, use the STIXM, SEM, and high resolution mass spec. Again, the idea is that if you look from left to right, day to night, and from larger particles to smaller particles, there is no significant difference here. So something very surprising, but important finding is that the particle chemistry can be independent sometimes, both particle size and the day and night time. And the very last one, I'll just briefly mention that you are also interested in the isonucleating particles. And one thing is challenging is to isonucleating particles, to identify the isonucleating particles. We're using, again, both SEM and STIXM, and to chemically map those isonucleating particles, because that's very important to understand, because these are very rare in the atmosphere. And then last slide, this is my last slide I want to briefly mention as the advancement in the field. So some of the studies that I mentioned about the in situ STIXM that we did, there was some work done by the Swiss White Source. They developed another microreactor, which is very advanced in a way that can have the gas flow, that can also control the temperature in a way that you can better control the humidity as well. And this is again another cool work done the same group with ice nucleation observation within the X-ray microscope, so you can get the ice nucleating particles in real time. And then finally, there's work led by Jay is under development at EMSL. He is trying to make a chamber where you can image particles under environmental atmospheric pressure, so we don't lose some of the volatile components. So these are some of the future direction that we're going to discuss during the discussion phase. I don't have to discuss now at this time. And then I'd like to thank all the folks that are doing all the work here. So thank you.